morning, students. Today's session we are dealing with the economic importance of bacteria and fungus. So let us see that you have already studied about the bacteria. It's a single-celled organism, means it is made with a single cell only. The whole life activities is carried by a single cell. And how about the structure? You can see whether any nucleus and nuclear membrane is present. No, this nuclear material only is present and no nuclear membrane is present. So it comes under the group prokaryote. Prokaryote means the organism which do not have a definite nuclear membrane. So we can say it's a prokaryotic organism, and whether it have any chlorophyll or not, no, it do not have any chlorophyll. So you can see that just like leaves, you can see any branches or leaves or stems or root, flowers, anything is present in this bacteria. No, it is not present. So I already told you it's a prokaryotic, means a single cell and no nucleus is present. And you can see that this bacteria, where it is present almost everywhere. You can see in our surrounding air it is present, in the water it is present, in the soil it is present, even on the surface of your body. Even on your cloth or on the surface of your body also these bacteria are present. Again, you can see that inside our body also, in the intestinal tract, even in the mouth also, you can see certain bacteria are present. And do all these bacteria are harmful to your body? No. You can see that some bacteria are very harmful. There are some bacteria are beneficial to us. Can you say any one bacteria which are useful to us? Yes, you know, in the milk, that bacteria which is present as lactobacillus, that is converting this milk into curd. If this bacteria is not present in the milk will not be changing into curd. So that is one simple example I told you. So some bacteria which cause many harmful diseases in human beings and even in animals as well as in plants. So these bacteria is present everywhere. And what may be the size of this bacteria, whether it is too big, can you see by naked eye or not? No, it is very small in size and you cannot see this bacteria with the naked eye. You can see this bacteria with the help of a microscope only, and its size is 2 micrometers long and 0.5 micrometers in thick. So you know it is very small in size. And coming to the shape of the bacteria, according to the shape of the bacteria, it is divided into 4 types. What are these 4 types? First one is Fungi. So first type, according to the shape, we are dividing this one. Second one is called as Bacilli. Third one is called as Spirillum. And again the fourth one is called as Vibrio. So these are the 4 different types of bacteria according to their shape. Now let us see the first one, Fungi. And this Fungi, it's of round shape or spherical in shape. These bacteria are of spherical shape. And whereas Bacilli, these are rod shaped one. And Spirillum, it is spiral in shape or spring shaped one. And Vibrio, the fourth one, that is of comma shape. So like this, it is comma shape. So according to the shape, you can say that these bacteria, 4 types you can see. First one is Fungi, that is what shape? Of course, here you can see it is spherical chain. Second one is called as bacilli, that is of rod shape, and third one is spirilla, that is of spring shape, and next one is vibrio, that is of comma shaped one. We will put comma now, that shape one. And again you can see most of the bacteria sometimes they live as a single one. Alone it can live. Whereas some bacteria they live together. Different methods is there. So you can see if this bacteria occurs in pairs, that is like this, in pairs together. If it occurs, it is called as diplococcus. So what is the term? It is called as diplococcus or diplococci. So, if the bacteria occurs in pairs, that is called as diplococci. And now, if it appears in the form of a chain, one after another, in the form of a chain, it is called as streptococci. So, if the bacteria appears in the form of a chain, that is one after another like this, in the form of a chain, it is called as streptococci. And coming to the third one, if it is in a cluster, that is together. If it is together, you can call it as what? Staphylococci. So, cluster, how? Like this, together. So, nature on the basis how these are living together. It is called as diplococci, then streptococci and staphylococci. So, what is meant by diplococci? Yes, if two bacteria, so the bacteria, if it lives in pairs, it is called as what? Diplococci. Whereas, if it is in a chain, in the form of a chain, it is called as streptococci. And if it is in a living in the form of a cluster, they are living together, that is called as staphylococci. So these are the three terms: diplococci, streptococci and staphylococci. Now coming to the structure of the bacteria. Coming to the structure, you can see that it is a simple, I already told you, it is a simple cell. Made with one cell, it is unicellular. And you can see that it contains a living substance called as protoplasm. So inside you can see, just I am drawing the structure of a bacteria. So this bacteria, this inside this together, including the nuclear material, the cytoplasm plus the nuclear material is called as what? Protoplasm. So this nuclear material altogether plus cytoplasm is called as protoplasm. It's a living material. And this contains, you can see that outside this one protoplasm there is a membrane. This is the membrane which is called as cell membrane. The inside membrane is called cell membrane. Externally one more membrane which you can call as cell wall. So it is double layered membrane. One inside one is called as cell membrane and the outer one is called as cell wall. And this cell wall, just like in plants, you know what is cell wall is present in plant cell, and with what material plant cell is made up of? Yes, it is made up of cellulose. But in the case of this bacteria, the cell wall is made up of peptidoglycan, a substance called as peptidoglycan, and it is a non-living thing. Okay, so this bacteria inside that, again you can see some of the chromatin material like this, a thread-like material, and one end of this chromatin material is attached to this cell membrane. So, usually in plant cell or in animal cell, you can see a definite nucleus with a nuclear membrane, nucleolus, and chromatin network you can see. Whereas here in this bacteria, you can see that just this is made up of DNA. This chromatin material only you can see. This is the genetic material of the DNA. This attached one end, it is attached to the cell membrane. And the cytoplasm contains, this is the cytoplasm, inside this there is a substance, this called thing we can call it as cytoplasm. Sometimes some granules like this, small dot like structure, some granules may be present in the cytoplasm of the bacteria. Again you can see in some cases, this externally to the cell wall, you can see a thick layer like this. External to the cell membrane, this is the cell membrane, external to the cell membrane. In rare cases, you can see a thick covering, that is this thick covering which is shaded, that is called as what? Yes, it's called as capsule. So, external to the cell membrane, you can see a thick membrane or thick slimy protective layer, and this layer is called as what? Capsule. And how do these bacteria move? These bacteria, most of them are passive, they cannot move by their own. So usually you can see that by the help of wind, water or by contact, these bacteria cells may be moving by with the help of wind, water or by contact. And in some bacteria, there are two long hair-like structures. Sometimes one hair-like structures, some other case two hair-like structures are present, and these hair-like structures are called flagellum. These from the top down. So flagella, both are together called as flagellum. And using this flagellum, two is the flagellum. So you can see that in water it can launch, just like how you are swimming like that. Using this flagellum, they can launch in the water medium and they can move from one place to another. So the locomotion, locomotory organ, generally we can say that there is a flagella. So actually it is passive in movement. It may not move by their own. With the help of wind, water or contact, you can see it may move from one place to another. And in some cases there are, you can see one or two hair-like structures which are called as flagella. By the lashing movement of this flagella, it can move in water. Okay, now let us see what is the mode of nutrition in this bacteria, how it obtains food. So, can you say whether these bacteria are prototrophic or heterotrophic in nature? Prototropic means one which can prepare its own food. What is meant by heterotrophic? One which depends on other things for its food. So, can you say whether bacteria is autotrophic or heterotrophic in nature? Yes, quite natural as it has no green pigment. It is heterotrophic in nature. Usually you can see that they will depend on the ready-made food from other sources. The different sources are there, so it will depend on the ready-made food from different sources. And according to the type of nutrition, how it gets its food, there are two types of nutrition we can see in bacteria. One is called as saprophytic nutrition and the other one is called as parasitic nutrition. So, first step is called as saprophytic nutrition. What is meant by saprophytic nutrition? Is the bacteria, if it obtains food from the dead and decaying material, that mode of nutrition is called as saprophytic nutrition. So, once more, if the bacteria obtain food from the dead or decaying material, that is called as saprophytic nutrition. Dead, decaying. Mainly from dead materials, if it get the food, it is called as saprophytic nutrition. What about parasitic nutrition? Yes, it is the method by which it obtains food from a host cell. That means host cell means what? The cell where this bacteria lives is called as host cell. Suppose the bacteria is in a human body. The human body is called as the host cell. If it is in a cow's body, the cow's body is called as the host cell. So, suppose this bacteria is obtaining their nourishment or their food or getting their nutrition from other living hosts, that is called as parasitic nutrition, other living host. So, these are the two modes of nutrition seen in bacteria. One is called as saprophytic nutrition and the second one is called as parasitic nutrition. Now, we can see what is the type of respiration seen in that one. Before that, this I told you these two types of nutrition, whether it is saprophytic and parasitic, how it absorbs these nutrients from others body or dead body or decaying body or from the host body, you can see. At the time of nutrition, this bacteria, it will release an enzyme into that food material, so that the food material, by the action of this enzyme, it will dissolve in this enzyme and then it will be absorbed by bacterial cell. That is a process taking place. Now let us see what is the type of respiration taking place in bacteria. So, respiration, you have seen that it's a catabolic process. It is breaking down of glucose we have seen. So, two types of respiration also you know. What are those two types? Yes, one is aerobic in presence of oxygen and the other one is anaerobic in the absence of oxygen. And again this anaerobic respiration, it is common in certain bacteria and fungi. So here in the case of bacteria, usually respiration is anaerobic, that is in the absence of oxygen. So here respiration is anaerobic. So, common method of respiration is anaerobic in the absence of oxygen. And these anaerobic bacteria means those bacteria which respire anaerobically. If it exposed to air, what will happen? Definitely it will be killed in presence of oxygen. It will be killed because usual respiration mode is by anaerobic in the absence of oxygen. Let us see the type of reproduction in bacteria. So, as it is a simple cell, bacteria consists of only one cell. It's a simple cell. You can see that by asexual means, usual method, you can see asexual method of reproduction. asexual reproduction, and one of the method is by fission. What is meant by fission? A single cell divided into two. That is called as binary fission. So here also you can see the reproductive method is by fission. You can see that this during the time of reproduction. So this is I am drawing. This is the first cell. That's a simple way. So this is the nuclear material I am showing like this. At the time of reproduction, you can see that the cell becomes little bit large in size, and this nuclear material it will duplicate. So second figure. So the nuclear material it will duplicate. So two it will be forming. So the cell expands and the middle a separating layer is forming in between the cell. So this on the third stage you can see. Like this, a small groove will be forming. A small groove will be forming, and this duplicated from nuclear material, you can see it is one will be moving to this side and the other one will be moving to this side. So the duplicated nuclear material will be moving to both sides of this cytoplasm, and finally you can say the next stage after this, the fourth stage, you can see that that is this groove it will grow again like this. Understood? Now understood how it is growing. So this will separate, it will become two bacteria. Each one can live independent life. Sometimes they will be together, each one with one chromatin material or DNA, like this it will be separated. This is a simple mode of reproduction you can see in bacteria, that is fission, especially binary fission. So here it is a simple way that the DNA duplicates, then a small groove will be forming, so that in between this cell wall is forming, just splitting into two. So the nuclear material will be passing from this to that. So this is a simple mode of reproduction. You can see that this bacteria, it divides for this division, it takes a very little time only. Within 24 hours, you can see this bacteria. It will multiply very fast and very fast. Okay, very fast method it will be multiplying, and a large number of bacteria can be produced within 24 hours. And still, I told you that this is a sexual mode of reproduction, and a very primitive mode of reproduction. You can see that is not common in bacteria. In rare cases, a highly primitive mode of sexual reproduction occurs. How this occurs, you can see that two bacteria of the same species or two strains of the same species, they will come together, and you can see that the plasmid of the donor, it goes in the, when they comes in contact with each other, you can see the plasmid of one of these bacteria, which is called as donor, goes into the other bacteria or the other strain, that is called as recipient, and this process is called as conjugation, and this is very rare case, and this conjugation we can say is a mode of sexual reproduction in primitive form only it will take place. And next we can see the spores, how bacteria will develop spores. Spore formation in bacteria during unfavorable weather condition. I mean, if it is too hot in drought season and all, you can see this bacteria. It forms we can see a hardened cover around itself, and that is called as what? Capsule. And these spores and all these cytoplasmic things will be inside that one. You can see it like this. Here the nuclear material. So this is what is spore formation. Then there is hard spore wall it will form. Here this is called as the spore wall. A double layered wall will be forming. And during this spore formation, this bacteria will be totally in an inactive condition. It can remain that dormant condition for some bacteria even for many years. It can leave this spores. And you can see that a hard protective wall called as spore. This spore wall is formed, and again when these bacteria gets or the spore get favorable condition, what happens? This spore wall will rupture or breaks, and it will get the normal shape of the bacteria. That is you can see to withstand the unfavorable weather condition. Bacteria itself got a covering that is called as spore wall, and the condition is called as spore formation. And when the condition becomes favorable, it breaks open or rupture this spore wall and comes out. Okay, and so this bacteria, you can say it can tolerate extreme dryness. So how usually you can say that extreme dryness means other organisms will die. Here, because of the formation of this spore wall and spore, it can withstand or it can tolerate this extreme dry condition, and it cannot be killed by the at a high temperature. Even not all bacteria, even certain bacteria, certain types of bacteria at a high temperature also it can withstand. We cannot kill that bacteria by applying heat. Then you can see even some bacteria they can tolerate poisonous chemicals also. Sometimes the other host cell may die by these poisonous substances, but even this bacteria will not be killed. Not all, even certain types of bacteria they will not be killed by poisonous chemicals. So three things that they can tolerate extreme dryness, they cannot be killed by a high temperature, and they can tolerate certain poisonous chemicals. So again these spores it is carried by wind or by water or by contact. These three methods I told you. It will be carried from one place to another place, and when this meet a favorable condition, you can see it from that it will come out. And you can see that protoplasm becomes active, and you can see that this nuclear material will be forming. So this spore formation in bacteria, you don't think that it's a type of reproduction. No, it is not a type of reproduction. It is only a thing to escape from the unfavorable weather condition. So in the different types of bacteria, different time period is for the this spore formation. So if they after getting inside the spores, some bacteria can live up to many years. So it is just a method to escape from unfavorable weather condition. Okay, now I am going to ask you few questions. Can you answer from the portion what I have taken? Name the common types of bacteria according to their shape. Yes, first one is cocaine, second one is bacilli, third one is spirillum, and the fourth one is vibrio. And these shapes Coccus, it is spherical in shape. Next one, bacilli, a rod shaped one. Then spirillum, it is spring shape. And the fourth one, that is vibrio, that is comma shaped one. Now the second is, and name the technical terms for these things. First one, if the bacteria that occurs in pairs, what do you call that one? Yes, that is Diplococcus. And if it occurs in long chain, it is called as Streptococci. And if it occurs in clusters, it is Staphylococcus. And what are the two types of nutrition in this bacteria? Yes, one is saprophytic and the other one is parasitic. What is the type of reproduction in bacteria? Yes, it is by asexual means and mainly by fission. And why do bacteria produce spores? Yes, to overcome unfavorable weather condition, bacteria produce spores. Now, let us see what are the useful role of bacteria in medicine. So, how you can see bacteria are used in medicine in the production of antibiotic, serum and vaccines. So, usually for three purposes these bacteria are used in medicine. One for the preparation of antibiotic, second one is for the preparation of serum and third one is for the preparation of vaccine. First, let us study about how these bacteria are used in antibiotics. So, you might have heard the word antibiotic. What is it? Yes, these are chemical substances produced by living organism which can stop the growth of some disease producing bacteria. So, antibiotics are chemicals produced by the living organism only, not by the dead one, by the living organism. And what is its effect? It can stop the growth or disease produced by bacteria. Such substance we are calling it as antibiotic. And do you know which is the first antibiotic found out? Yes, that is penicillin, commonly used in medicine. Examples of antibiotics, you can see streptomycin, it's an good antibiotic. Then chloromycetin is another antibiotic, and tetracycline is another antibiotic. So, you can see the four different antibiotics: streptomycin, chloromycetin, tetracycline and penicillin. And penicillin is produced, the first antibiotic produced. It is from Penicillium notatum, that is a scientific name. It's from a fungus. So, the fungus name is called as Penicillium notatum and it is discovered by the scientist, you can say it by Alexander Fleming. So, Alexander Fleming discovered this antibiotic, that is penicillin, from Penicillium notatum, and it is a product of a mold or fungus and not of bacteria. And streptomycin, you can see, was discovered by Salman Waxman, and it is in a similar way a source of organism was Streptomyces griseus. And what is the criteria for a good antibiotic? This antibiotic you can see for we are getting for some certain diseases. And what should have this? What are the quality of a good antibiotic? And you can see that this antibiotic, it should be able to kill a variety of microorganisms. This antibiotic should have the capacity not only to kill one microorganism, to many microorganisms should be killed by this consuming, you can see some side effects we may get. So a good antibiotic should not have any side effects. And the third one is it should not kill normal bacteria of the host cell. So I told you that in human body itself many useful bacteria are present. So when we are consuming an antibiotic, this normal bacteria should not be killed in our body. So thank you.